All right. So unfortunately, what happens is the second day there was a little bit of technical glitch again. Uh, I initially was just doing some small talks and just started the Zoom, and I just paused the recording and I just forgot to resume the recording. So just only last 15 minutes of the lecture was recorded. So well, I do have an integrity, and I promise that every lecture will be recorded, even if it is five, six people, even one people if need to see this because they were absent or due to some reason or something like that. I still have to record it, but hopefully we'll just get out of these issues in a week. We will get used to it. Anyway, so we stopped at this slide, and what we are seeing in this slide, we talked about that the diploid cell, the advantage of the diploid cells, which are not cancerous cell in making the viruses, because so that the oncogenic DNA doesn't get into the along with the virus, not delivered if a vaccine is made into some of these transformed cell line, which are aneuploid, sometimes do have oncogenic DNAs. Another important thing is that if you just need a virus in a large amount, several milligrams, 100, 200 milligrams, or in a gram amount, I'm like, how do you grow that the large amount of the virus? And first of all, and also why you need those. So sometimes to grow a large number of a virus, there are several different types of things that is needed. First of all, there are vaccine production stains that is required by the pharmaceutical companies, which all viruses are not made in cell culture. There are several different things, but some of them are made in cell culture. And also you need it for structural biological studies to make crystals of the virus. You need a large amount, lots of amount of virus. So when you do this, you can use some of the cell line which can grow in suspension. That means that they do not need an adherence. Here, the cell lines that you are seeing, primary or the continuous cell line, they always stick to the bottom of the flask or the Petri plate, and they are adherence dependent. Now, if you need to take them off, what you do that you add them like an EDTA, and this EDTA is a chelating compound which actually mops up the calcium ions which are present in the media, and that basically inactivate the adhesion molecules like integrin, which actually basically responsible for attaching the cells in there. So they inactivate all these adhesion molecule. We call this CAM, or cellular adhesion molecules. They inactivate those if you take the calcium out of them. Other than that, also we need to use that along with the EDTA, either a trypsin or chymotrypsin, sometimes collagenase. These are proteases, and these proteases are needed basically so that some of these non-inactivated adhesion molecules or the cellular adhesion molecules or the CAMs are just slightly digested so that they can come off. So once you just treat the cells a little bit, then you can scrape the cells out pretty easily. Otherwise, it's very difficult for them to come out. And then you can passage the cells, and immediately you dilute them with the large amount of, you just centrifuge the cells down after they come off. Then you take the supernatant off and dilute them with a regular media so that you take off the EDTA and dilute the EDTA and the protease in such an amount that there is no remnant. They can stick back in there as well. So these are some of the way that you can just move the cells. But the suspension cells don't stick at all. So say, for example, the lymphocytes, which move around in the blood, the white blood cells, they do not need any media to adhere to that. And also, very interestingly, the insect cells, there are several ones that is derived from many different types of insects. Very popular is some of the caterpillars insect cells. They are large and they are not adherence dependent, and you can grow them in a large flask, like in the laboratory, 500 ml, 1 liter, 2 liter flask with a magnetic star bar, and sometimes you can scale them up in the industry up to say to 100, 200, 500 liter scale, and you can produce a vaccine out of those. So classic example, the Shingrix, the vaccine for the shingles, is actually made in insect cells, basically derived from a caterpillar called Spodoptera frugiparda SF9 cells, and these cells are infected with a baculovirus. This baculovirus is an insect virus, and these recombinant baculovirus have the whole sequence of these surface of these not the papilloma, the varicella zoster virus, which is the particle of your chickenpox virus. So the chickenpox virus is caused by a varicella zoster virus, and what you do that you produce a kind of the virus-like molecule by inserting some of these proteins that is needed into the baculovirus. The baculovirus goes into these cells that does not kill the cell because it's recombinant. That's the lethality of that is gone, but they produce the proteins which assemble spontaneously and make VLPs, or the virus-like particles. These are the vaccine for your shingles or the Shingrix, because your cells, when you inject it, understand that that is the surface protein of the varicella zoster virus and raise antibody against them. And again, we're going to talk about those in later amount. So these are some of the usage of the viruses in the cell line. Now, along with that, there are several different areas of a chicken egg that is highly used for vaccine production as well. And you can see that there are several different areas: chorionic allantoic membrane, amniotic area, yolk sac, allantoic inoculation. There are several different areas, and different viruses grow in different areas. You do not have to just remember the different areas of the egg where the viruses can grow, but you do not have to correlate with those. Just take one example though. See that the influenza virus grows in the amniotic fluid, the amniotic cavity right here around the embryo. On the other hand, you can see that right here, this is the allantoic cavity. This whole largest space is the allantoic cavity. Influenza virus also grow in there. So in the industry, the influenza virus is produced in the chicken egg, and they are grown in this allantoic cavity. So we're going to see that in the next slide, that these are the robotized material where one row of a chicken egg can be inoculated as quick as 10 seconds. So the robot actually drills a tiny hole in there, inoculates the virus, seal it back, and you get the virus. The virus is basically produced in the allantoic cavity. Remember that where the viruses are growing, these allantoic cavity is basically lined by cells. So remember that it is not growing in a media or suspended in the media, lined by the cells, and the viruses are growing in there, and they just come out in the allantoic cavity. From there, they are harvested. Sometimes you can see that when you just go for the influenza vaccine, sometimes the pharmacist asks that, do you have an egg allergy? If you being asked that question, you know that particular vaccine, the influenza vaccine, has been manufactured in eggs. Nowadays, there are several different types of recombinant vaccine that is being made, and also now they are made in recombinant plants. You get these virus particle produced in the plant product, like a tobacco plant, and you just harvest the leaves, crush them up, purify the virus-like particles of the viruses, which are the vaccine strain, basically, and you inject them for the vaccine. Usually there is in an influenza vaccine, there's a mix of 4 of them. I'm going to talk about that somewhere else. So these are some of the interesting features. Now, when you infect the cells, what you observe in many cells is what is called a cytopathic effect, or this is the pathology of the cells. So what you are seeing in this picture, the top are the normal cells in a phase contrast microscope. And when you infect these cells with a virus, over the period of time, you can see that there was some changes that will happen in here. You can see right here, some of the cells basically are rounding up more and more. So over the period of time, the normal cells, basically, you can see that produce these round refractile cells, and they are gradually basically dying. So in the first picture right here, all these were adhering to the bottom of the Petri plate or the flask or the wells. And then gradually the cytopathic effect here will make the cell round, and they will also lose the attachment or the adherence. And this is one type of cytopathic effect, or the killing effect of the virus. Now remember that the cytopathic effect, intensity of the cytopathic effect varies highly among the viruses, and some of the
some of the viruses will not even show cytopathic effect. They can infect the cells, they will produce the virus, but they will not kill the virus immediately. Sometimes we call this as a persistent infection. They will allow the cells to live, and that cells will start make the virus for a very, very long time. So on the other hand, there is another type of thing that can happen, that formation of a syncytia, syncytium singular, syncytia is a plural, where the virus is responsible for fusing the cells. And here you can see that this virus does have a marker, blue marker, and here this cell actually do have some recognizable antibodies or receptors on top of that. And when you have these two types of cell in there, and if they're both infected by the same by the virus, which can fuse or infect both the cells, you can see that in the syncytium you have the both markers, the red and the blue, become now or present on the same cell, and it is multinucleated. So here we are looking at, let's take a look at this for a second. So if we just look at the picture of the cells, you can see that these are the round cells which are showing the cytopathic effect, and they have a round refractile ring around with that. But what we are looking at, that right here, if you just look at this cell, this is a giant cell right here, and you are seeing right here, these are the nucleuses which fuse together. See here, you have another giant cell in the background. On top of that, there are some of them which are floating in there. But here you can see that this is a nucleus, right here, nucleus. Here you are seeing another giant cell right here. So you can see that these are the viral factories made up by the virus. These are floating cells though. And you can see that the nucleus is right here. So see that these giant cells don't have a refractile edge like those. What is the difference? This is a phase contrast microscope. These giant cells are still sticking at the bottom of the Petri plate. That's why they do not have a refractile bottom. On the other ones are floating. They are at a different level, or they're focusing in a different level, and that's showing that shiny ring in the phase contrast. So because they are suspended in the media rather than sticking to the glass. So this is a typical phase contrast microscopic picture. So let's take a look at that. All right. So this is your syncytia formation. Some of the viruses do that. Say, for an example, Sendai virus is one of the examples. Cytomegalovirus or CMV is one of the types, herpes virus. These are some of the viruses which actually do form these syncytia. Not all the viruses does that. Some of the viruses do that. Build these large factories. It is much more efficient because you get a large amount of cytoplasm and lots of nucleus to drive the production of the viruses. So here you are looking at different types of cytopathic effects. So definitely know the morphological alterations and what we mean by implosion bodies. We're going to explain that. And the specific examples are not required. So you do not have to remember both the lines, but just remember that what are the different types of cytopathic effect. Say morphological, nuclear shrinking, proliferation of the membrane, nuclear membrane, vacuoles in the cytoplasm, syncytia, we already called that, which is cell fusion, margination and breaking of the chromosome, rounding up and detachment of the culture cells. So we actually saw these pictures of these, and also we saw the syncytium formation. We have seen these two at least in pictures. On the other hand, inclusion bodies means that there are some dense structures like inclusion-like structures in the cytoplasm of the cell. These could be the virions in the nucleus, virions in the cytoplasm. And the classic example, definitely remember this one for your exam and the quiz, that right here in the cytoplasm, you have these Negri bodies. These are dense structures which are produced by the rabies virus. This is a classic example that for a long time, these Negri bodies are the typical cell that if a person does have rabies or an animal have rabies, if you just take the cells out and look at them under the microscope, you will see these Negri bodies in there. So on the other hand, there are different type of guanine bodies factorized in cytoplasm by the pox virus, clumps of ribosome, clumps of chromatin in the nucleus by the herpes virus. These are some of the inclusions that you can visualize in the cell under the light microscope. You will not be able to see the virions, but when there are many of them together, you can see a darkly stained electron dense area in an electron microscopy, or a dense area if you stain the cell as well. So these are some of the example of the cytopathic effect. So after that, we are looking at the assays of the virus and how you measure the process of measurement of the virus in the sample. And it could be two types. It could be either a physical, seeing through a microscope, or biological means of the viral assay, which is the infectivity assay. And the virus titer is known as the measurement of concentration of the virus in the sample. And remember, these two words are very important. What is the type of virus assay? There are many different types. And the virus titer is that if you are given a sample, like a viral stock in a vial, and you just want to know that, okay, all right, in that vial, how many viruses are there which are infectious? And that will be the virus titer in there. So for an example, you just probably used a different type of word that sometimes we check for antibody titer, whether you have taken a vaccine and whether you still make antibodies or not. So at that time, you check the antibody titer. That means that your blood is being drawn and it is checked for the amount of antibody present. We call this as an antibody titer. So just remember these wordings. So several different assay by biological methods, and we are just going to discuss one at a time. So 1952, this Renato Del Becco actually developed the plaque assay, and it's revolutionized virology. And it is a classic assay. Many viruses do produce these plaques, but again, remember that not all viruses produce plaques. You have to measure them by different means, but many viruses produce plaque, and it's a very, very elegant assay to assay the viruses. And he also received Nobel Prize in 1975 for a different type. We're going to talk about that. He actually could transform cells on the Petri plates as well by the RSV, or the Rous sarcoma virus, and it just paved the path for many other different types of discoveries. We're going to talk about that. So the plaque assay is one of his greatest discoveries, Renato Del Becco. And now we are looking at the plaque assay in the visualization, that what you are seeing. So what is a plaque? So here, suppose you have a Petri plate, which is covered by a monolayer or a single layer of cell, which is 90% confluent. Usually we use the cells when it's 90% confluent, means 90% of the surface area of the Petri plate is covered by the cell. The reason for that, as you are adding the virus and the virus is taking time, which is the eclipse period, to produce the virus out of the cell, by that time, that 10% of the area, if needed by the cell, will be covered up. Why we do this? The reason is that all these adherent cells grow by a process which is called grow, but they do have a property which is called the contact inhibition. If the cells don't have a place to grow, if they touch each other, then at that time, what will happen? They will not pile up against each other. They will just float up and start dying very quickly. And you will not have a proper plaque assay, because you will have lots of cells in the suspension floating around in the media, and you will not get a proper assay. So all the cells must be adhered to the bottom of the cells. That's why we start with a 90% confluent wells. If you have a 50% confluent, on the other hand, most of the areas are empty and you are not going to get a plaque. So that's why you need that. So what happens in the cell, that we just infect them with a virus, and the virus gets into one cell, one virus particle per cell, one cell in an ideal condition. You can argue if two virus particles can get into one cell. Of course it can, and we're going to see and address that. Once this virus particle gets into the cell, it will replicate, and the new viruses will come out bursting the cell, and it will infect the neighboring cells. These neighboring cells will now die, and the more neighboring cells will be infected, and the plaque will grow until a point where you can see them under the light microscope. So here you are looking at, in a phase contrast unstained sample, On the other hand, there are some of the plaques which cannot be visualized just by other method, by the naked eye. You can use something with a recombinant virus. So not all viruses are recombinant. So if you are just doing this with a non-recombinant native virus or the wild type virus, you cannot do that. But for the experimental purposes, we make recombinant viruses many times. And how we do this? In a recombinant virus, you take out some of the genes which are not necessary for the virus to function. And you replace those genes with a gene which is called a LAC-Z. And this LAC-Z is a gene of the LAC operon from the E. coli. And the LAC-Z produces a code for an enzyme which is beta-galactosidase, which breaks down lactose into glucose and galactose, or the milk sugar. That is the function of the lactose utilizing enzyme in the E. coli. And you put them in the virus, and now in the media in the plate, what you do that you have a chemical which is known as X-gal is added. Now X-gal is a substrate just like the native lactose of beta-galactosidase. So if there is a plaque formation like this, where you are producing recombinant viruses in the plaque, the virus is going to produce that LAC-Z product out of the gene, which is the beta-galactosidase. That's going to digest that X-gal or split it up, and it produces a color. So once the X-gal substrate is digested by the LAC-Z product, beta-galactosidase, it produces a chromogenic substance, blue color, and you can see these blue spots. These are good if you're just working with a recombinant virus, basically, and if you cannot see the plaques properly. So how exactly it happens? On the other hand, this is a video microscopy where you can see a plaque formation in a time-lapse videography. You are going to see that these viruses are producing a green fluorescence protein. So you're going to see that there are some green cells that you are seeing. The cells are not green, the cells are colorless. But as the viruses are being produced, these on the background, it is an ultraviolet light is being shown on top of that, and you will be able to see this green fluorescence protein in the background as well. So let's take a look at this. So here you can see, just to take a look at the middle of the cell. These are almost all confluent. These are some of the debris that floating around, but just take a look at that. There is a green cell right here and something happening in here. See that the cells are gradually clumping up, and now a plaque started to form, and it's increasing in size. And these cells, you can see that these are all green, means that these are filled with recombinant virus producing that green fluorescence protein. And it is an amazing video that you can see that how a plaque is formed in a video microscopy. Lots of these cells which are showing the cytopathic effect, they're all tearing up basically, and that's the formation of the plaque. So a fantastic picture. You can see that how it is so cool. All right, so let's take a look at the assay method. And I have just written these assay methods in very detail in the note. So this is similar to the serial dilution experiment that you have done or heard in the microbiology. You do this with the E. coli experiment, but the serial dilution experiment to find the concentration of E. coli in a sample. So here what we start with, that suppose you have a virus stock, and you take 0.1 ml of this virus stock and add in the test tube having 0.9 ml of virus stock. So here you are just adding 0.1 ml to 0.9 ml, so the volume becomes 1 ml. So out of 1 ml, 0.1 ml is the virus. So your virus concentration, or the virus, is diluted in here 10 times. So 10 to the power minus 1 is the concentration of the virus. Now you take 0.1 ml from here and put in another 0.9 ml. So here you have 10 times dilution here, 10 times dilution here. From the first to this is 10 to the power minus 2, or 100 times dilution. And then 1,000, 10,000, 100,000, then you have 1 million, and then you have 10 million fold dilution from here. So you just serially dilute that. Then you take, sometimes you may have to go even to 14, 15, minus 10 to the power minus 14, 15 samples, that if you have a very, very concentrated viral sample. Then from these dilutions, you usually do not use the very concentrated ones because you're not going to get a proper result. You take 0.1 ml of sample, 0.1 ml is a 100 microliter sample, and then you add those to the Petri plates with 90% confluence cells that we just talked about right here, 90% confluence cells. You allow the virus to attach or adsorb for approximately 1 hour. You just add that inoculum, leave it there, and then what you do that you remove that 0.1 ml by washing the cells gently with some plain media without any viruses. So all the virus which were suspended in the media, they are out. You have taken them out. And then, so you remove the inoculum. Then you overlay them with a molten agar kept or equilibrated at 50 degrees Celsius. When you add them onto the cells at 50 degrees, it's not going to kill the cell. Or you can use the carboxymethyl or CM cellulose, which both of them are gelling agents. So it's going to form a gel on the top of the cell. Now remember that when you make this agar or the CM cellulose, you use the media or the cell culture media with them. So you use, say, for 50 ml of agar, you add to that and you dissolve that into say 50 ml of agar, well, say about 2% agar, and you add 50 ml of 2x media. So in 100 ml, the media concentration becomes 1x. Again, I'm not going into the detail, but just know that there are nutrients in there as well. So you overlay them and they will solidify, and then you just incubate in the incubator at the permissive temperature, which is usually for most of the animal cells, 37 degrees Celsius. And the plaques will form as quickly as 48 hours or 2 days. Sometimes in some of the cells, it may take even 1 or 2 weeks, those depending upon that how virulent that virus is or that plaque formation characteristic is this. And different cells will produce plaques at different rates. And some of the viruses will not produce plaque at all. That's also possible. So then what you do that after that, you will stain the cells and you will count the plaques at the very end. So what the function of the agar or the CM cellulose is, and this is a quiz question, exam question, definitely remember that when you add these semi-solid agar or the CM cellulose on top of the cells, it keeps all these viruses which are coming out of the cell in the same place. If they are with the media, like a liquid media, the virus will just basically diffuse in the media. And suppose you have a foci right here, and it can just go somewhere else and infect some other distant cells. But when you take out the liquid media, no liquid media, you have a semi-solid media, the virus which are coming out of one cell, infected cell, will only be able to infect the neighboring cell, not jump into some other areas. And this is important so that the virus doesn't travel much distance, just remain in next to the, or remain in the cell, or just able to infect the neighboring cell of the original cell. That's the function for the agar or the CM cellulose. And then you just count the plaques, and suppose here we have taken the sample from a 10 to the power minus 6 dilution well, and you have taken 0.1 ml, which is 0.1 ml is 10 to the power minus 1. So 10 to the power minus 6 plus 10 to the power minus 1, you just add them up. So you get 10 to the power minus 7 is the final dilution of that. So if you count 17 plaques in there, the concentration will be 17 multiplied by 10 to the power minus 6 plus 1, 7. And that's equal to, you just express that as a 1.7 into 10 to the power 8. 17 into 10 to the power 7 is equal to 1.7 10 to the power 8. You just move the decimal right here. This is the plaque forming unit in 1 ml of your virus stock. Remember that what you did, that you multiplied the number of plaques with the number with the dilution factor, and the dilution factor was 10 to the power minus 6 plus volume plated of 0.1 ml. So minus 6, 10 to the power minus 1, total 10 to the power minus 7 was the dilution. You got 1.7 or 17 plaques. You multiply the dilution factor with that and get the original count of the virus. So you back calculate that. Now, what is a plaque forming unit? Plaque forming unit is basically the virus which is infectious in the cell to form one plaque. And we'll define that much more clearly later. Now, if you just see the kinetics of the plaque formation, this is called a one-hit kinetic, means one cell, if it is infected by a one virus, you can going to get a straight linear curve like this, and the number of plaques is directly proportional to the power of the concentration of the virus inoculated. So you can see the relative virus concentration, and this is the number of plaques. These are directly proportional. So as the concentration virus is double, the number of the plaques will also double. So in this condition, this type of graph you get when there is one cell, theoretically, one cell is infected by just one virus. But you can argue that if two viruses infect one cell or three viruses do that, what happens? So if it is a two viruses do this, then you get a two-hit kinetics where the number of plaques is directly proportional to the square of the concentration of the virus inoculated, and you get this type of graph. So this was also discovered in some plant viruses, which does have two sets of genome. When you have these two sets of genome, they're independently packaged. And when the plaques used to be formed, the scientists were puzzled by the numbers that they are seeing in that. And from there, they found that they calculated these two hit kinetics, as if the one cell is infected by two individual viruses. Now, there is an important thing that what we do is the plaque purification. So once these plaques are formed, before you stain them, of course, for visualization, you stain them, but you do a plaque purification to clonally select a virus. What do you mean by that? We assume that one plaque is formed or started with one virus, one type of virus infecting one cell. Now, you know that viruses mutate in the cell all the time. So what you do, that assuming that that virus is one virus infected one cell and they are all the same, you usually plunge a micropipette and take out a small amount of this agar or the CM cellulose plug. Basically, you just suck it up, and then you just put that agar plug into a media. And the viruses, whatever you sucked up from those plaque, is going to be suspended in there. Now, this is your virus stock. From here, you do a serial dilution and do another round of plaque assay. Then in the second round, you again pick up a plug of agar, suspend that, and do a third time. Usually when you do this plaque purification at least 3 times, you usually get a clonal virus stock. That means each and every virus in that stock theoretically have the same genetic makeup. The reason that we do that, as the viruses constantly mutate, when we are doing the experiment, we just want only one type of genetic makeup of the viruses. Sometimes, if you have some of the cells infected by 2 viruses or 3 viruses, you can get a mixed population. So from a mixed population, we buy the plaque purification technique, you get, instead of a mixed virus stock, you get a clonal virus stock where all of the viruses are the same. That's the function. So here are some of the other type of assay, other than the plaque assay, that what you do is that this is a fluorescent focus assay. So here, this is a modification for non-plaque forming viruses. Here, either the virus can produce a recombinant green fluorescence or red fluorescence or any type of fluorescence protein that you can measure under a confocal microscope or a fluorescence microscope, or sometimes what can happen that you have an antibody which can recognize a virus antigen. So say, for an example, I'm just giving a random example. Suppose you have a SARS-CoV-2 and you have the spike protein and you have antibody against the spike protein. So you have a cell culture which is growing the SARS-CoV-2 and you add these primary antibody which is against the spike protein, and it will bind to that. Then what you do that you use a secondary antibody with a fluorescent dye attached to it, which is commercially available, that will bind to the primary antibody, and you can visualize these under the microscope. If you have taken cell biology, understanding this is a little bit easier. If not, just look at the immunofluorescence, that how it is done. Just understand the basic principle, you will get that. And here we are looking at that this is the dilution, and here you are looking at the fluorescence of the basal cells. So that's basically you're measuring in an arbitrary unit. You're just measuring the fluorescence. This is fluorescence focus assay. On the other hand, this is an assay, which is, we call this as an infectious focus assay. So here is a modification of plaque assay. Here, we are not looking at the viruses that is present in the viral stock, but we are measuring what fraction of the cells in a culture is infected with a virus. So how we actually do. So here we take a known number of cells, basically. So say for an example, we take 10 to the power 6 cells, 1 million cells in a small test tube. And then we infect it with the virus. And after we infect with the virus, we just allow them to infect for a certain period of time and then centrifuge down the cell and take out the supernatant or remove the media. So all the viruses which are in the supernatant, not inside the cell, they are out. Now, we don't wait too much for the cells to lyse. We just allow the virus to get absorbed. We use the word absorb, A-D, not A-B, not absorb. Absorb means that when a virus or any molecule basically touch or just come in association on the superficial structure. So the viruses basically is attaching and they are being then pulled inside the cell. So viruses are absorbed and delivered into the cell within that time, and we just harvest these cells. Then what we do that we take a plate where you have already a monolayer of culture. You already have some cells present in there in the 90% confluency, just like your plaque assay experiment. On top of that, you add these infected cells with different dilution. So you take these million cells, and then you dilute, you take 0.1 ml of that sample and dilute it with 0.9 ml of media. Then from that, you take another 0.1 and dilute it with 0.9 ml of media. And you get 10 to the power -1, 2, 3, 4, 10 to the power 6. These are the different dilution that you are actually seeing. Or what you can do that either without the dilution, you can increase the number of the cells as well. So you can just take, say for an example, 1,000 cells, 100 cells, 1,000 cells, 10,000 cells. You can vary the number of the cells that you are adding on top of these cells. Depending upon that, either you can dilute or you can just increase the number or the fraction of the cell. So depending upon that, what you are seeing right here, you can see right here about 10,000 cells of that whole fraction of say 1 million cell. They are producing all these plaques, means that they are producing. This is basically the infectious focus assay where you just produce. This is the correct assay for the plaques, and you can just measure that. That is that whatever your virus stock is. If you use 10,000 cells, you are going to get a plaque assay. That's basically what you understood. If you use less amount of cell, it wiped out everything. You can see that this is controlled uninfected. But if you have 10 to the power 5, 6, or 7, you get very little amount of plaque. So whatever concentration of virus you have, if you use that virus for your particular cell, you will take 10,000 cells that will infect and form the plaque. So here you just trying to understand without measuring the virus, how much percentage of the cells that you can use with that virus to measure its infectious focus assay. The next one is a transformation assay, and this is only possible to do this with the oncogenic viruses like Rous sarcoma viruses. So here in this Rous sarcoma virus is one of them, which basically transform the cells into the cancer cells. And what happens that in the background cells, what you are seeing that these are all adherent cells, means that they are attached to the bottom of the well. And they are, here you're seeing confluent cells, but they grow by contact inhibition, means they will stop growing at contact inhibition, means that when the cells are 100% confluent, there is no space for the cells to grow, they will stop growing. But the cells, when they become cancerous or when they are transformed, the cells can pile up on top of each other, and you will see these focuses, which we called as the transformation focus or foci. And this is where the cells are growing on top of each other. In the phase contrast microscope, that gives you this different contrast. So you can measure these oncogenic viruses by this transformation assay. After that, we are looking at another assay, which is called an endpoint dilution assay. You can do this in the cell culture, just like here, the picture is in a 96 well plate, micro well plate. But you can do this in animals as well. You can just do this in mice, in rats, rabbits, even larger animals. In there, you just need a large amount of them. So basically what you do, that you take the virus stock and you dilute the same way as the plaque assay, -2, -3, 4, 5, 6, and so on. And then if it is a cell culture, you add those viruses in duplicates in these rows of cells. So what we're seeing, that if you take in 10 to the power -2, you see the cytopathic effect in every cell. And these cytopathic effect, how do you see that? You just see these under the microscope. And these cytopathic effect could be one of these. So here, if we just go right here, that what are the cytopathic effect? It could be one of these. You can see the virions in the nucleus, or nuclear shrinking, or some of the proliferation of nuclear membrane, or negative bodies, or clumps of chromatin in the nucleus. You can see some of these cytopathic effect. So when you see that pathology of the cells in here, you just mark them that, okay, cytopathic effect observed, and you can see that at this very high concentration, 10 to the power -2, all of the wells are showing cytopathic effect. As you dilute, it started that some of the wells may not have a virus, and you are not seeing the cytopathic effect. So if we take that 10 to the power -5, you can see that you have 1, 2, 3, 4, 5 cases, you don't see any cytopathic effect. Whereas 1, 2, 3, 4, 5 cases, you see cytopathic effect. So your 10 to the power -5 concentration is known as the tissue culture infectious dose 50%, or TCID50. ID50 is infectious dose, TC is tissue culture. So what it means, that the 10 to the power -5 viral dilution in a cell culture for that particular cell will be able to infect 50% of the cells. So this is known as the endpoint dilution assay. And you can see that if you go above that, -6 or -7, right here, you do not see much cells infected. You see just only one well, which is infected with a virus at 10 to the power -7. None of the wells are infected by the viruses. So this is also another type of assay that you can do. So now, one of the important concepts that we need to address from the plaque assay, we talked about this, that a particle to PFU ratio, and we talked about the PFU. So what is a particle? A particle that in a cell, when the virus is produced, every particle that is being assembled is a virus particle. Now, unfortunately, what happened, that not all the viruses that come out of the cell, either by bursting or by shedding by the process of budding, whatever the process is, some of the viruses may be defective. They can sustain damaged virus particle. They may have mutations and complete loss of replication, or there could be complexity of the infectious cycle, and they are not very successful. In some of the cells, they may not properly do. So what happens, that usually in a viral sample, when you have a sample that you are measuring, there are usually most of the times more number of virus particles than infectious virus particle. So all the virus particles in a sample is the virus particle. And the plaque forming units are technically the virion. That means those are the ones which are the infectious virus particle. So the PFU is a plaque. Why we use instead of virus, we tell the plaque forming unit. Plaque forming unit is the virus which are able to infect the cells and produce the plaques. Not all particle will do that. So this is also important, and we can calculate the the particle to PFU ratio, the number of physical particles of infectious particles. And you can just do that by electron microscopy. You look at the number of particles in a sample, and then you just look at the PFU by your experimentally. You can just take a look at that. And let's see, the number varies hugely. So here, right here, the papillomavirus, see that the particle to PFU ratio is 10,000. So you can just easily imagine that the large amount of papillomavirus that is produced is defective. It varies, and you can see that pox virus, it is in between 1 to 100. Whereas, just remember this one, all of them you don't have to remember, but this alpha virus, Semmeliki forest virus, is a classic example of this alpha virus. Is the particle to PFU ratio is 1 to 2. What does that mean? That means, suppose you have produced 100 particles in a cell, one physical particle over infectious particle, if the ratio is 1, 100 out of 100 particles is infectious. If it is 2, then 50% of that, 50 particles out of 100 is infectious. Still, it's remarkable. So you can just easily understand that why it is important. So on the other hand, if you see that this number, pox severity is 1 to 100. What it means, if it is 1, that means again 100%. That means that out of 1, all of them are infectious. On the other hand, if it is 100, then out of 100 viruses, there is none that is infectious. So if you take 10,000 virus particle, 900 of them will be non-infectious, just 100 of them will be infectious. So that's how it is. So for SARS-CoV-2, it is 2,000 to 1,000. So for SARS in the mice, it's 67 to 540. So here, if you look at the data, you can just tell, it's like scientists always lie. Why it is 200 to 1,000? Remember that there is a lot going on, and these are different reports. Why they vary? It's not there is a scientific error. Remember that we are trying to grow these cells, all these viruses, in many different types of cell. Some of the primary cells, some of the lung cells, some of the nasal turbinate epithelial cells, or some of the cell lines like Vero cells, BHK cells, 3 cells, 3T3 cells. These are the cell lines, and all these cells vary in their property. And in some of the cells, the virus is very successful in making the particles, and at that time, you may get a lower number like 200, 300, 400. In some of the cells, due to the condition of the cell, the virus is not that successful, and it makes a lot of mistake, and it just doesn't produce that many infectious particle. Many damaged particles are produced, and you get a larger ratio like 900,000 like that. So that's the reason that depending upon the condition of the cells, this varies. So this is a very important concept. Definitely remember that. The next, we are looking at some of the viral assays by the physical method, and let's go one at a time. So the first one, we are looking at the electron microscopy and the fluorescence microscopy, or the super resolution microscopy, just to directly visualize the cells in the viruses in a sample. Now remember that when you do a transmission electron microscopy, classical transmission electron microscopy like this, usually we do take a negative stain. You can definitely underline that word or write that word. We use a negative stain which stain the background like this, and you can see the contrast, the virus against that. This is a bacteriophage. So somehow we look at some of the discovery of transmission electron microscope, and we saw some of the negative stain slide in the lecture topic 1, but there is a question about this as a negative stain, but here we are also emphasizing that. So definitely remember that your transmission electron microscopy, classical transmission electron microscopy, will need this negative stain. If you do a cryo-EM, which is a more recent technique, transmission electron microscopy was discovered in 1942, whereas a cryo-EM is more recently. Here, you use just water to freeze your sample. For a classic transmission electron microscopy, you have to take your sample in the virus, suppose it's in the cell, then you embed in a plastic block or epoxy block. You just cut sections of that, and then that section you stain that with the negative stain and then visualize. With the cryo-EM, you do not have to do anything. No staining is required. So you take your sample in water, just plain simple water, and you flash freeze them in liquid ethane or liquid nitrogen, which is minus 160, or for liquid nitrogen is minus 190 degrees Celsius. You plunge that the sample into that. So basically what you do, you take a loop on which you put on in the electron microscope. You had the sample in a water on that grid, and you just plunge that loop into the water. With the sample, you plunge that in liquid ethane or nitrogen, and the water instantly freezes. And this type of when you freeze the water that fast, it gives a unique property of water. Remember that when ice is formed, the ice is crystalline. It has a crystal structure, and this crystalline ice is very sharp edges. It's going to completely destroy a cellular structure or the viral structure. They have very sharp edges. But when you instantly freeze water, there is no time for the water to form this ice crystal lattice. They freeze in an amorphous condition, which we call as a vitreous ice. So in the vitreous ice, it is glass-like. See, the glass is amorphous. Let's say, for an example, you take the sand, which is silica. When you just fuse them or just melt them, basically you form basically an amorphous glass, and this is the vitreous ice, or the amorphous ice is basically that. You have a glass-like structure, and then you shoot them with the electron beams, and you take the images in different planes by tilting them a little bit, and from that you calculate the structure of these viruses in the cryo-EM. The scanning electron microscopy you can visualize mostly the infected cells. Direct virus visualization is almost impossible because the scanning electron microscope the resolution is not as high as a transmission electron microscope. Scanning electron microscope only visualize surface areas. So cytopathic effect can be visualized very, very well, or some of the virus which are making some Negri bodies, or some of the other type of cytopathic effect can be visualized, but individual virus particles is just probably a little below the scanning electron microscopic level, except some of the giant viruses. Also, if you have another very powerful technique, is the viral particles are now tagged with the fluorescent proteins or the dyes, or antibodies attached to fluorophores of the virus expressing the fluorescent proteins. All of these can be exploited to see the viruses under the fluorescence microscope. And some of these fluorescence microscope is a super resolution microscope, which is a modification of these microscope. And you can just see a single viral particle as a fluorescence particle. So you can just easily see this is light microscope. Remember that. In a light microscope, you cannot see a single virus particle by staining. But when it produces a colored signal, that can be observed even from a single virus particle. That faint signal can be observed by a fluorescence light microscope or the super resolution microscope. Here, what you're seeing, the red structures are the microtubules. And on the microtubules, we are visualizing basically movement of some of these virus particles on the microtubules. And on the microtubules, we're going to see somewhere else, there are motors which carry these virus particles through the microtubule from one part of the cell to the other. These motors are dynein and the kinesins, which basically attach the virus particles and move on the highway inside the cells, which are the microtubules. And you can visualize them. So here we are looking at another very important experiment, is a hemagglutination assay. So some of the viruses, and we are looking at here influenza virus, other than that, there are several different types of virus. It can agglutinate or it can clump the red blood cells. So how this is basically done? So there is a protein called hemagglutinins on the surface of many of the viruses, including influenza viruses. So hemagglutinins is your H. So you can see that we talked about the H1, N1, H5, N1. So your H is the hemagglutinins. And what are these numbers? 1, 2, 3. These are the different types. They are all hemagglutinins, but their sequence varies a little bit. That's why you have these 1, 2, 3, 4. These are different types. N, on the other hand, stands for neuraminidase. That's an enzyme. which basically cleaves a glycoprotein, which is a N-acetylneuramic acid, NANA, or the sialic acid. They can cleave those. So that's N is a neuraminidase, an enzyme, and that is also present on the surface of the or the envelope of the influenza virus. All right, but N is not responsible. It is. We're going to talk about that in a second later. So what happens that the hemagglutinin is only responsible for that. So here what you do that you just add the viral particle with the red bloods, and it's going to clump the red blood together, which we call as an agglutination and hemagglutination, clumping of the red blood cells. So what we are seeing right here in these microwell plates, you dilute the virus, say 1:4 all the way to up to say 1:8192, 8,000 times. And then you take these red blood cells and you put the red blood cells, a certain amount of red blood cells in each of the well. The top A row, you have not added any virus. So these are virus free. So when you have virus free sample, what happens after adding the equal amount of red blood cells, all these cells, they are heavy. They are full of hemoglobin right here. They tumble at the bottom of the well and they form this sharp dot or button. So these are the unclumped free red blood cells which tumble down at the bottom, forms this. But now what happens in the row B is that you have added virus in this dilution 1:4 to all the way up to 8192. In this different dilution, you have added the red blood cells and then added the virus along with them to agglutinate them. So what you are seeing that say from 1:8 all the way up to 512, you don't see a dot. You see a diffuse reddish color. So what is happening that the virus is doing the agglutination of the red blood cells, and they are forming a lattice-like red blood cells, and they're floating in the media. They are not going to the bottom of the cells. They're just diffused throughout the media. That's why you do not see a sharp dot. But what happens that from say after 512, 1024, 2048, and these two wells, you are started seeing the dots appear back again. Why you are seeing the dot? These samples have too less amount of a virus to do the agglutination reaction. It's too less. They are not being able to form that lattice, and that's why the red blood cells are dropping towards the bottom of the well. Too less of an amount. So here, if you just look at the hemagglutination index of this virus, from your stock, 1:512 dilution will be the hemagglutination. This is the threshold that you are getting. Now here you have a question that if you just look at the 1:4 sample, you do see a dot. Why do you see this? So this is very interesting. So very carefully understand that these viruses, along with the hemagglutininin, which hemagglutinate these viruses, also contain the neuraminidase. That's the N. H is the hemagglutininin, N is the neuraminidase, which is the enzyme. Now, the hemagglutin, the hemagglutininin that you have on the virus, that binds to a surface receptor on the red blood cells, which is the N-acetylneuramic acid, also known as a sialic acid. So when they bind to this sialic acid or N-acetylneuramic acid, NANA, they bind to one of those with one of them hemagglutininin, then another hemagglutininin is bind to another NANA on the other cell, and then another one to the other cell. That's how it clumps. But neuraminidase cleaves these cells, these N-acetylneuramic acid or sialic acid, they cleave them. Why N is in there even. So what happens that when influenza virus comes out of the cell, during the budding process, to get it out of the cell, it steals the membrane, and the cleaving process, the way it can egress or exit the cell out of the membrane, is that they digest that attached protein, which is the N-acetylneuramic acid, and that helps the virus to release. So the neuraminidase functions in the exit of the virus or budding of the virus out of the cell. That's the only function there. Now, in a high concentration, it digests the glycoprotein, which is the N-acetylneuramic acid or the sialic acid, and now you do not have any of those at a higher concentration. So the viruses cannot bind onto the surface of that because they are all cleaved by the neuraminidase, and they tumble at the bottom. That's why at a very high concentration you appear the dot dot appears. I have just wrote that clearly in the notes, so you will get that. All right, so after that, we are looking at viral enzymatic activity. So here what you are looking at, that some of the reverse transcriptase activity of some human endogenous retrovirus is being measured. We produce RT. We do have reverse transcriptase function in there. Remember that these endogenous retroviruses do produce some of the protein and the enzymes, but they cannot form a full particle in them unless you are infected with a live virus, like you have HTLV or HIV positive. You do not have the live viruses integrate or functional viruses integrated into your genome, but you have the HERV with the human retroviruses. We talked about that many times. So these are different type of HERVs, and we are looking at the retrovirus. We measure the activity of them, how much of the reverse transcriptase they produce natively in the cell by the assay of the enzyme. So what you are doing right here is a polyacrylamide gel, where what you do that you just give them a primer, which is an RNA primer. Remember that what is the reverse transcriptase reaction? The RNA is converted to DNA by the reverse transcriptase. So to that you need a primer. So you give them a primer, and that primer is going to be converted into the DNA by the reverse transcriptase. This is happening in the test tube. So what you're seeing that right here, you have two samples of GST HIV RT and the AMV RT. These are two control reaction, two different types of reverse transcriptase. These are commercially available. And what you do that when you add them in a reaction mixture, what the reaction mixture does have, it has RT. You have an RNA primer on which it's going to convert it to the DNA, and then, or the template rather than a primer, I should say template. You have the template, and then what you have that you have the nucleotides, DNA nucleotides. So you have DNTPs. So DNTPs means that DATP, deoxy adenosine triphosphate, deoxy so you have DATP, DCTP, DGTP, and DTTP. These are the deoxynucleotides. Out of that, usually either the DATP or the DGTP is labeled with the alpha 32 phosphate. So the phosphate is radioactively labeled. So when you form a product or the DNA from the RNA to DNA, you are going to see a band in the polyacrylamide gel. So all the free nucleotides, which are really low molecular weight, you are going to see them at the very bottom of the gel. But when they're forming a product, you can see these products are higher molecular weight. These are the DNAs that is being produced, although very little, but you can see that these are some of the DNAs products that has been formed. On the other hand, here you can see that how these products are being formed. See that you see this ladder-like, this is a ladder-like structures right here. And these are the reverse transcriptase DNA products that's being formed in here. And you are measuring them in a polyacrylamide gel for 3 different viruses. So what exactly you're looking like and why you would have this ladder-like structure. So remember that you are doing this experiment in a test tube, and these enzymes are not very robust or anything, and they fall off the template. Say for an example, here the enzyme, as it is making the DNA from the RNA, goes up to say 450 base pair and falls off. The next, another enzyme probably went another 20 base pair more, went to 470, then fell off. Maybe another one went another 10, like 480, and then fell off. So due to those things, that the different length of these DNAs are formed from the RNA template. And that's why you are seeing these ladders, and this is a typical RT assay that you can see. So depending upon that, you can see that the RT activity of this 7.1 and 10.1 are almost similar. Right here, the data from here is plotted. They're almost similar. Whereas for the 10.9, it produces much less amount of product. Here you can see the product is much less. So here the RT activity versus incubation time over the minutes, you can see that the different enzymatic activity of the endogenous retroviruses. So you can tell that the endogenous retrovirus RT activity of the 10.9 is much less compared to 7.1 or 10.1. So you can just measure the RT activity for some of the viruses. All right. So after that, we are looking at some of the enzyme-linked immunosorbent assay, or ELISA, which detect the viral antigen or the antibodies. So here we ask two different questions. The first, that whether you make any antibodies against the virus or not. It could be two scenarios. Either you are infected with say SARS-CoV-2 and you want to know that whether you have antibodies, or you can ask that you have taken the vaccine, whether you have still antibodies or not. So here your blood will be drawn and you capture these antibodies and put them on a solid support in an ELISA well. And you have the commercially available viral antigen. Suppose you have the viral antigen as a S protein or the spike protein, and these are produced commercially in many different areas, and you just have the viral antigen. You add the viral antigen, and then you have a secondary antibody you add, which also detect the viral antigen, but it is added with a fluorescent dye or indicator. And if you see that there is an indication, you know that you have the antibody. No dye means there is no indication. So this is one of the direct ELISA, or the enzyme-linked immunosorbent assay. On the other hand, you can also do a rapid test where you are asking the question, are you infected with the virus or not? Whether you have the virus. So this is a typical test that you do, the saliva rapid test. You take your sample and you have commercially available antibody against the virus antigen, say your spike protein. You commercially have this. You add this antibody sample, which is the immunoglobulin, and if you have a positive result or a positive sample, the antibody will bind to it. Then you add a secondary antibody, and this secondary antibody is the anti-immunoglobulin G, IgG antibody. And remember that the primary antibody is the human antibody, whereas the anti-IgG is usually raised against this in, say, a goat or in llama, large animals, or even they are grown in mouse or rats. But usually commercially, the goat or the cattle or the horses, llamas, these are the ones which are humanly bred to have these secondary antibodies. Once they are purified, these antibodies are attached with this particularly chromogenic substance or the indicator. Usually there is an enzyme called horseradish peroxidase that is added to these, and you add a substrate. If there is an antigen antibody binding, and if you have the viral antigen, then you will get a colored reaction, which you can read at an ELISA plate. Whereas the interesting part is that the lateral flow test that you do at the point of care test is based on this as well. So basically what happens that if you have a sample, a positive viral sample in there, these are wicked, so when you just put them on a test strip, the wicking action of the viral antigen will pull it, and if you have a viral antigen, it may bind to the antibody. And then these antibodies, they are going to be further pulled. And if you have a positive viral antigen, it's going to bind to an anti-IgG line, and it can give you a colored reaction in there. So you're going to see a colored reaction on the test strip. If you don't have a viral antigen, then it's the antibody sample. Nothing is going to be wicked on the antibody sample. So this is not going to move anywhere, and you're not going to get any indication. Sometimes you can get false positive results in there as well. It's not a very robust test, whereas the ELISA, when you do it in a plate, is much more sensitive than the lateral flow with the wicking action, which is a modification of this ELISA test. All right, after that, we are looking at the viral polymerase chain reaction, and this is an extremely powerful technique, where in this technique, what basically you do, that you take your DNA sample, and you can amplify it many times. So one copy after 35 cycles of PCR, you get 1 billion copies of that, because what you're doing is like 1 to 2, 2 to 4, something like that. So you get just very few amount of sample, and you need the DNA template. Then you need to know the sequence of that so that you have the primers, and you mix with the nucleotides. and you need a thermostable DNA polymerase, which is the Taq polymerase, which are basically from these thermostable bacteria that Thermus aquaticus originally, and then we produce them recombinantly now. So in a classical, let's take a look at, so in a classical experiment, what we do in a classical PCR experiment, you actually run a gel. So in the run a gel, suppose you have a lane 1 and you have a lane 2. So what you do that when you run the gel after a PCR, in one sample, you don't have the template or you don't have the sample. So that's your lane 1. There is nothing on the lane 1. You should see a clean lane. On the other hand, say for example, the gene that you are amplifying, suppose it is 1500 base pair. And suppose this is your 1500 base pair or 1.5 kilobase right here. That's your marker. So suppose this is a molecular weight marker. You run that. And then what you should have that if your sample is positive for that diagnostic, you are going to get a band right here. So which is 1.5 base pair. So if you see that band, that means that you're positive for the sample. So this is to do this, usually you have to run in between 35 to 40 cycles of PCR. Then you purify the product and then run on the gel. This will take a very large amount of time and almost impossible to do that in a scenario like COVID testing RT-PCR. So we have a modified product and we're going to see that what we are going to do in this way. So let's take a look at that what we are going to do with these. The first thing that the DNA viruses, you can directly, PCR can analyze the nucleic acid. For the RNA viruses like SARS-CoV-2, you cannot amplify the RNA with the DNA sample. So you have to have a reverse transcriptase first to convert that RNA into DNA. That is your RT. After it is converted to the DNA, you do a PCR. That's why it's RT-PCR. Reverse transcriptase is the first reaction. It's in all reaction mixtures at the same time. You had the reverse transcriptase, Taq polymerase, everything in one. But the first step, the RNA is converted to DNA, double-stranded DNA. Then you do the PCR on that. That's your RT-PCR. The second is that the PCR that you do is not this type. But instead, what you do that your PCR nowadays, we call this a real-time PCR or quantitative PCR or qPCR. So what you have that one of the nucleotide, there is a fluorescence chemistry or many of the nucleotide, they are fluorescently labeled, not radioactive, but fluorescently labeled. And in the PCR machine, there is a laser light source. As the test tube, transparent test tube is producing the product, the laser light is constantly shining and there is a detector. And in real time, you are getting the product formation, information of the product formation. So if you just have the panel of that, and I'm just changing here the color of the pen. Say for an example, you are looking at a product like this. Then you have another sample where you are looking at a product like this. So you are seeing this, and basically you start a product formation, and right here, both of them look the same, but you may have a product formation. Say for an example, I'm just trying to get another color, say magenta. You are seeing like this. So you can see that right here you have an initial lag phase, and then you have an initial lag phase right here. So you can see that right here, this is the level, which is the threshold level where the fluorescence goes above the baseline. But what is happening here, this basically gives you the overview of the number of cycles that is required. So suppose these are the number of cycles for this. This is the number of cycles. These are the number of cycles. So for this first sample, suppose that you are getting the product after 10 cycles of the PCR. So your cycle threshold or the CT value is 10. Here is also 10. But say for an example, this is about 20. Sometimes you can get a CT value of 40. So how you interpret the data of the CT value? So if you get a cycle threshold CT value about 10, you have a very high confidence to say that yes, this is a positive sample. And here it is a little bit low, but probably will say yes. If you get a sample after 40 40 cycles of the PCR, you get a positive result. Yeah, you can tell it a positive, but probably everything in a sample, like you take any piece of DNA, you will get after 40 cycles or 50 cycles, you will get some product. So highly chance that your CT value, if it is 40, that they are very unlikely that you have a meaningful virus out there. Probably it is false and minus. So the diagnostic company basically determines a cutoff point. Say for an example, if and again it is a trade secret depending upon the kit. It depends that for some samples or some kit, maybe 15 cycle threshold is positive. Some maybe it's 25. That means that if your CT value is 15, then you have a positive result. If you have a CT value of 25, you have a positive result. But if you have a 26, 27, chances that you are a negative result. So depending upon that, you can just identify that in real time. You do not have to run a gel or anything like that. You just run hundreds of samples together. You can just do in a one PCR machine, you put 300 tubes and you can just run this experiment 300 samples at the same time. And you can just get these data in an extremely fast manner. And you can just go in a high throughput way by analyzing a large number of data in a short period of time. So this is the power of your nucleic acid, basically. So that's about it. So next, what we are going to see is the PCR product is not the same as the infectious virus. And right here, I realized in the class that probably I have not turned on the recording button. So I did not resume the recording, and here I actually found that. So from here onwards, I am just stitching the class real lecture with the previous one, and that will take care of it. So here we are looking at some of the samples, and we are looking at that the PCR product is not the same as the infectious virus. So you can see that this is an example with the Zika virus. I did not put in the SARS-CoV-2, that this is the infectious Zika virus infectious Zika virus particle a mouse is producing. And the RNA is detected in the sample after a long time. And you can see that on the other hand, this is a vasectomized mouse. This is they collect the sample from the semen. You can see that this is the infectious virus, whereas the RNA is still available for a longer period of time, shedding in seminal fluid. So you can just easily imagine that if you are doing the RNA, they are available for such a long period of time. So you can question then, okay, all right, so if I'm taking a nose swab, how this is going to work and tell me that I have COVID, I don't have that one. The reason is that usually in that area, if you have some RNA, that means there is some virus in that part. But interestingly, what happens that if you just go into your blood sample and draw some blood, you still may have some RNA in there for a certain period of time. Particularly, it is also known as the long COVID. Some of the patients get that long COVID, and there was a maximum that we actually got in a Chinese patient after she was asymptomatic in that process. They found RNA, the SARS-CoV-2 RNA, up to 90 days, 3 months after post-infection as well. So that does not mean at that point that person is infectious. It is just the fragments of the RNA that is floating around into your body and will degrade over the period of time. So you have to take the sample from where it is actively replicate, like your nasal terminae, and from where it can go to some other person. If you get an RNA from there, chances of that you are positive and infectious at that time. But you may find the RNA from the other part of the tissue fluid or the body fluid, or even lungs. If you just sample out from the lungs, you may find RNA. So that's the important thing, and very, very important at this. We're going to discuss that as well. On the other hand, think about that we're taking the environmental samples from the sewer water, and we're just trying to identify that.
That gives you an overview of that. That does not mean that you are finding RNA in the sewer water in the Clemson-Pendleton area. That does not mean that those are virus particles in there. Remember that the virus particles, particularly the SARS-CoV-2, they are extremely fragile. They are just going to just get destroyed. But the RNA, they're still hanging around a little bit, gives you the idea of that in the community, how much virus are in there. And so also another important thing is remember that the viral RNA is covered by a nucleocapsid, usually a protein. And over the period of time, that comes off. And also we have the RNAs everywhere, in your palm, everywhere. So if somebody is working in a lab in the RNA, they know that. And this RNA is in the environment, in the sewer water, everywhere. And over the period of time, these RNA that are being dumped in there is going to completely get digested. So if you have a high level, that means that RNA is fresh, not hanging out from 3 months old. So that's why the data is from the sewer water, gives you a very good idea about the community prevalence of the virus as well. All right, so here we are looking at some of the burst or the definition concept that this was done by the Max Delbruck, and he found that a cell, particularly a bacteria, approximately 150 bacteriophages actually come out of the cell. And even some larger eukaryotic cell, it may be 1,000 to 10,000 virus. And is not observed in many viruses because they shed the viruses by budding. And these are usually the enveloped viruses, as we said, over the period of time. But some of the cells, like say, for example, polio, they will just synthesize all the viral particle and they will burst the cell, come out. And this was first studied by the Max Delbruck in T-event bacteriophages like T2, T4, T6, other T-events. So what they did, basically, it is these are the things that what we are talking about, that the experiment is in detail in your notes. So they just add these viruses to absorb onto the bacteria, then remove that, dilute the culture immediately, and then you take the time points of the sample and assay the amount of virus that you are seeing. So what they found that we have talked about, that this is a typical bacterial growth cycle. They did not find this, but instead we already talked about this, so this is repeat, that they found that after this diluted the virus actually disappear for some time. This is called the eclipse period. So what happens that at this time, even inside the cell, there is no infectious virus particles. Where did they go? At that time, the virus injected the genetic material into the cell. So the genetic material is hanging out, but there is no infectious virus particle at that time. So they completely disappeared out from the inside the cell. Then they assemble all together and they just come out exponentially. So this is where if all the cells are infected in a synchronous fashion, this is what it is. On the other hand, if you have a very dilute sample, you can see the multiple burst means at the first burst, some of the cells have been infected. These viruses are coming out, and then they are infecting the rest other cells and the rest. So suppose you have 1 million cell and you put 10 virus particles in there, you are probably going to see this number. So just remember that these are important quiz question. Think about that. If you have right here a larger number, say 1,000 cells, and you have just put 100 virus particles with 10:1 ratio, you are going to infect all of them. But on the other hand, if you just say that you take 1 million cells and you take 2 virus particles, you are going to see a multi-step growth curve. So here what we are talking about is a single and a multi-step bacterial growth curve for 2 different types of viruses. The first one basically kill the cell by bursting. So you first dilute the virus, and just to make sure that there is a correction in that cell, that if you just have the previous version of your PowerPoint, it was written right here under the total, it was written as intracellular. That was a typo from the book. So if you have that one, I just corrected that slide. So I just took the image from the book, but that it was wrong in the book. Sometimes that happens. So this curve is for the extracellular. Then you have the red one for the intracellular, and then the total is the purple one. It was written as intracellular as well. So after you inoculate the virus, you have an eclipse period. And in the eclipse period, the virus totally disappears. And after that, what you're seeing that in between the eclipse period and sometimes you just find extracellular virus as well. This is a latent period. So what the latent period after the eclipse period is, see that in the after, very carefully understand that, after the eclipse period to the latent period, so in between these two dotted lines, you are finding intracellular virus. They have assembled and they are about to come out of the cell. And at this point, the second dotted line, they have burst open the cell and you find extracellular virus. Before that, they were formed. Here, they are forming all the components, the genetic material, the proteins, and everything. Here, they are formed inside the cell, did not come out yet because their number is gradually need to increase before they can make the cell burst. At this point, the cell burst, come out, so you can see that both of them exponentially rise. The second example, on the other hand, is a Western equine encephalitis virus. This is a budding virus. And at this virus, you can easily see that as it buds out of the cell, you have more of a parabolic growth curve. You can see that this is the total or extracellular virus. This one is extracellular, that's total intracellular virus. Here, you do not see a very sharp growth curve. The reason is that here the viruses are coming out over the period of time. So you can see that to some extent you get a exponential growth and then it plateaus out basically because it's shedding at the constant level, just not that sharp. So budding viruses versus the cells, the viruses that actually basically kill is a little bit different. So now what happens is that to achieve this, we need to infect the cells, but how we will know? So now we are introducing another term, which is known as the multiplicity of infection, the number of infectious particle added per cell, or the PFU, or the number of cell. Now, this is not the number of infectious particle each cell receives. Say, for example, if you add 10 to the power 7 virus particle to 10 to the power 6 cells, MOI of 10, each cell does not receive 4 virions. And this is explained by what we call as a Poisson distribution. You just take some of these buckets and you throw these tennis balls, dump it over all of them. They're not going to receive all of them at the same time. So that's the thing, the probability that the Poisson distribution basically gives you. And here, this is the statistical distribution, that PK is the fraction of the cell, that is e to the power minus m, and the m is right here, is the MOI, k over factorial k. So when you make the k 0, fraction of the uninfected cell, if it's 0, it just becomes e to the power minus m, and m equal to 1n p0. If it receives 1 particle, p equal to 1, this is me to the power m. And fractions receive more than 1 particle, it becomes this. So if you just, again, you do not have to too much go into that. We'll ask a very simple question out of that. If you now calculate the P0, say for an example, 10 to the power 6 cells with an MOI of 10, if you just remember that E is the Euler's number from there, this 45 cells are going to get infected out of 10 to the power 6 cells. Remember that, 10 to the power 6 cells, only 45 infected. Now, the cells receive 1 particle is 450. So you can see that out of a million cell, only 450 has been infected. And on the other hand, the cells that receive more than 1 particle is 0.9995. Therefore, the culture of 10 to the power 6 cells, 999500 cell, or 99.5% cells are infected. So if you calculate now the M or the MOI, 1N equal to 0.01, you calculate that it is 0.4.6. So that's pretty much it. You will see that this is the example of that, and we will stop here.